Welcome back, friends. In this video we'll be talking about point mutation. This is a kind of DNA mutation, or you can also call it a DNA point mutation, a long term. But actually point mutation means there is a mutation in a single base of the DNA, because mutations are of different types. For example, if we take an example of a DNA, let's say A, T, T, G, C, T, A, other strand of the DNA should be, T, A, C, G, A, T. So this is a DNA, for example. If you look at this DNA, there can be a single mistake. For example, a problem with incorporation. Let's say here they place a instead of C. So here it is a point, it is a kind of mutation, right? This is a type of mutation, and this mutation, as the mutation occurred in one base only, it will be termed as point mutation, because the mutation takes place in only one point in the DNA. That means in only point here means base. So point mutation means the mutation occurs in single base pair or single base in the DNA. Okay, so there are many different varieties of point mutation, because the point mutation can be of different types. It can be during the production of the mutation, or also different natures of the mutation. The point mutation can be many different types, and the example of those point mutation are different types. Examples are, nonsense mutation, missense mutation, there is a frameshift mutation, and there are many more. Frameshift mutation contains both of the type, like addition mutation as well as deletion mutation. That means addition of one base or deletion of one base, that's also considered as a point mutation. There could be a substitution, so ultimately there could be a substitution, or there could be addition or deletion. So these are the different ways to get a point mutation in. So means mutation is a single base pair or single base in the DNA, and there can be another type of mutation. They are not point mutation. For example, if the mutation occur in the stretch of nucleotides altogether, they won't be termed as a point mutation. So these are the different types of point mutations, and in this video we'll be talking about each of those point mutation in much more details, starting from missense, then nonsense and frameshift mutations in much more details. So stay tuned and watch this video for all these details. So let's talk about different types of point mutations that we see in our body. Point mutation means the mutation in DNA and actually in one particular nucleotide. The change in one particular nucleotide will change the protein expression, because remember what we have DNA as a central dogma. From that DNA we produce mRNA, from that mRNA we produce proteins, right? So what happens here, if any nucleotide change occurs in the DNA level, it will create some change in the mRNA level. So the change in mRNA level ultimately reflect in the protein level, because that mRNA will be translated into proteins, right? So point mutation means the mutation in a single nucleotide. Mutation in a single nucleotide in the DNA, and that will change. That can be in the mutation in the DNA level, that can be mutation in the mRNA level, and finally ultimately reflect on the proteins. Because what we know about codons. Codons are 3 nucleotides long stretch. For example, CAT, this is one particular codon which is responsible for producing a particular amino acid, for adding a particular amino acid. In this case the amino acid is histidine. So if there is a codon called CAT in the DNA level, it will incorporate histidine, right? Or if it's an mRNA level, CAT, it will encode histidine. So that histidine is now added. Similarly, for different codes, different amino acids will be added, and ultimately those amino acid will create the primary structure of polyamino acid or polypeptide sequence. Say histidine, proline, serine, arginine, they are linked with each other, they produces a kind of primary structure. Now that structure will be folded to make the secondary structure and tertiary structure of the protein, that is the actual functional unit of the protein. Remember, none of this primary structure of proteins are actual active or functional, because for a protein to be functional it should be folded, and protein folding is still a big mystery. How it's done and all these things, theories are there, but it's not that much clear. But the protein folding is required to have that protein ability to function properly, right? Now what happens here, during any kind of nucleotide change in this, for example, instead of T there is a G placed, that will change this codon, or for example, instead of this A there is a C placed, that will completely change the amino acid which will be entered inside. So if we change the amino acids, remember there are 20 amino acids, they have different properties. Some of them are hydrophilic, some of them are hydrophobic, some of them have a bulkier group like proline, some of them are very tiny like glycine. So those are different types of amino acids and required to produce different types of proteins. For example, for producing membrane protein, we need to have more of a hydrophobic amino acid stretch to produce that hydrophobic core which will be embedded into the membrane. So during that production, if by any means of mutation we add more and more hydrophilic molecules there, that protein will no longer function as membrane protein. So these are the examples of why this mutation causes such a big difference, right? So if you look at here, for the missense mutation, so let's talk about the missense mutation. In the missense mutation, for example, we take the example of simple element like CAT, let's say CAT, 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 CAT. For simplicity's sake, we just have a very basic structure. Yes, CAT, CAT all along. Now we know this CAT is very easy to memorize too, because if you all time need some example to draw and write, I believe you draw this. CAT means cat, and cat will produce histidine amino acid instead. Remember that. So cat, 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 this is a stretch of nucleotide sequence in the DNA, for example. Now what happens here, for example, during this stretch, somehow they change one of these bases. For example, this particular base got changed. Instead of A, there is a C incorporated. Rest of the things remains the same. CAT, 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 CAT. So remember, this is the DNA normally. After the mutation, the cause of mutation is always the same in different cases. For example, it can be misincorporation of the nucleotide during the DNA replication. It can be misincorporation of nucleotide during the transcription, that is making mRNA from DNA, right? So these things always present. Or during the different kind of physical or biophysical and chemical agents like UV radiation, there will be production of thymine dimers like that, right? So these things are always related. For example, during this process, this A, instead of A, during the DNA replication process, the polymerase mistakenly put C instead, and the DNA repair mechanism will not recognize that, they pass this. So ultimately that mutation remains there as a C instead of A. So what it will do? What it does actually, the cat, this means histidine, histidine. So histidine, histidine. The previously we have all of them were histidines, right? All of them were histidines. But now these are histidines, we all know. But during this mutation, these 3 stretch nucleotides are termed as the reading frames, right? So rest of the reading frames remains the same. But the reading frame where we inserted C instead of A will change the amino acid to be incorporated. And they now incorporate proline instead of histidine. And proline is an amino acid with bulgier ring that may cause certain problems. So ultimately, instead of histidine, 5 histidine stretch of amino acid, we get 2 histidine and proline and then 2 histidine. So this, for example, this previous protein will fold something like this after this process of translation. But this protein, as a result of this proline, steric hindrance is caused due to the bulgier loop, and protein folding is not possible. So ultimately the misfolded protein will be guide to the protein degradation. So we will have a loss of activity of the protein. or it might produce a malfunctioning protein which does not function what it should do inside the cell. So this thing can occur, and that is called the missense mutation. Remember, missense mutation means missense. Sense means the sense of the codon. Miss means they miss the sensing of the codon. That means the codon should be histidine, but they miss the sense and produces proline. That is called the missense mutation. Okay, and that missense mutation can produce defective proteins which may not function properly, or that can produce proteins which are not folded, will be degraded inside the cell. Okay, now let's talk about nonsense mutation now. This is another type of point mutation. That means there will be a change in a single nucleotide there in the DNA, in the either DNA or in mRNA, which will lead to the production of faulty proteins or something. But in case of nonsense mutation, they produce a truncated protein. Why? Let's look at here. For example, here, let's say we have CAG as a stretch of nucleotide sequence for our understanding sake. We have CAG stretch. Okay. Now the CAG codon actually codes for the amino acid glutamine. Okay, so they produces glutamine in all these cases. So we have glutamine, or let me actually change this to black color, because so they all produces glutamine. Right? Now let's talk about the mutation. Due to the point mutation, by any means, what they did actually, they produces a T instead of a C there. For example, here in this C, instead of C, they substitute with T. So what happens actually, everything remains the same, all this protein reading frames remains the same, but here we have a T instead of C. So we have this stretch. Now what it does actually, CAG, remember CAG stands for glutamine. Glutamine is added during the translation now, production of proteins. CAG, that means glutamine is added. CAG again, glutamine is added, another glutamine is added. But now instead of a CAG, here we are having a TAG. But a TAG means during the translation, tRNA once recognize this TAG presence in the DNA, that means it should be UAG in the mRNA. Remember, because in the mRNA there won't be any thymine, instead of thymine there will be uridine, right, uracil. So in that case they recognize UAG. UAG acts as one of the stop codons in the whole codon format, in the genetic code, right? So there are 3 stop codons, UAG, UGA, UAA. UAG is one of them. So once they recognize UAG there, instead of DNA we are having TAG here, that means it acts as a stop signal for the ribosome. And the ribosome and the all protein synthesis machinery will dissociate from each other, and that will halt the synthesis of the protein. So ultimately the protein should have a 5 stretch of glutamine in the normal condition to function properly. Instead what it will produce, a 3 stretch of glutamine, which is much shorter or truncated protein. Now the truncated protein can have different problems inside the cell. This might have complete different function from this original protein, right? And there are examples of that. That's why I'm telling. This truncated protein may not function at all, and it should be degraded by chaperone inside the cell, by the proteasome complex and ubiquitination complex. This truncated protein can add with another truncated protein to form different variety of proteins inside the cell. So we never know what kind of thing is going to happen, because these are all examples of real-time examples that we have seen. So this truncated protein may have function improperly, or the truncated protein function like the earlier protein, but not all the functions that should be carried out by that. So many varieties, many problems can arise for this type of mutation. And remember, it is called nonsense. Actually it's nonsense because it actually makes a lot of sense that it will not produce any proteins, but the sensing of codon is completely blocked. That's why it's called nonsense type of mutation. But in missense type of mutation, there is no truncated protein. Protein will be produced, but there will be different amino acid placed, right? So that's it. So alright guys, let's talk about frameshift mutation. So that was the gateway of silent mutation. We have already talked about that. We have talked about silent mutation, missense mutation, nonsense mutation. So I will encourage you to go back to the videos and watch those videos because it will be helpful for you. Now in this video I am going to talk about the frameshift mutation. Frameshift mutation. So in the frameshift mutation, what we want to say is, again we'll take the simple example of, let's say, CAT, stretch of CAT nucleotides present. Okay, so stretch of CAT nucleotides, and the CAT nucleotide, CAT is a codon, you know. So for understanding frameshift mutation, you need to have the understanding of genetic code and codon. So codon means 3 nucleotide sequence which acts as a signal to add a particular corresponding amino acid during the protein synthesis. For example, during protein synthesis, what happens? This is the mRNA. So mRNA contains all those, say for example U in mRNA, so all those structures here, all those nucleotides, and there is a ribosome sitting onto that, and there are tRNAs which will detect the presence of each codon. So codon means 3 nucleotide, for example CAU, these are the codon. So there is a tRNA which also have an anti-codon loop exactly to bind with CAU, right? So it should have, you know, GUA. So this tRNA with GUA can actually pair with CAU, which is in the codon present in the mRNA. So that tRNA will go and attach there. So here is the tRNA attaching here, and that tRNA is corresponding to, there is a site for amino acid binding in tRNA, and that tRNA will bind to a specific amino acid corresponding to the CAU codon, and here the amino acid is, you know, histidine, right? So histidine corresponding to CAU will attach with this tRNA, histidine specific tRNA, because there are specific amino acid tRNAs present in cell. So that tRNA will bring histidine to the place. So on, depending upon different codons, different tRNAs will bring corresponding amino acids, and they will join the amino acid, then produce the proteins. That is the simple way of protein synthesis explanation. Now, so once we know this concept of codons, so for 3 there will be a particular defined tRNA with defined amino acid. So for CAT, all these cases we should put histidine, histidine, histidine. So we are having a kind of track here, kind of 3 tracks here, 5 amino acid track, all of them will be histidine, right? Now what happens in frameshift mutation? This each codon during the transcription and during the translation process, the protein synthesis process, are acting as a open reading frame. They are called as open reading frame or ORF. So each ORF consists of 1 codon, and 1 codon means 3 nucleotide sequence, which is here CAT. So this open reading frame kind of fixed. For example, for normal case, it is CAT, 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 and each open reading frame will insert 1 histidine together. Ultimately, they will add 5 histidine by translating this DNA. I mean, translating this mRNA which is produced. Okay. Now during this process, for example, if by any means of error during the DNA replication or transcription they add an extra nucleotide in somewhere here, or they delete one nucleotide from here. So let's see. Okay, so now this is the reading frame. These 5 reading frames are there. Now let's say during the process they insert a new nucleotide which should not be placed here, but they during the DNA replication or transcription of mRNA they add a nucleotide there. For example, they place all these reading frames firmly, but here they place one extra adenine in this place, right? So that is added here. So once this thing is added, what it does actually, it increases the number of nucleotides. Previously there was 15 nucleotides, but now there is one extra nucleotide which is A added. Now remember the difference between this frame shift with the missense and nonsense mutation, because in those mutations we never see an addition or deletion, we have seen the substitution. For example, instead of A there is C, or instead of T there is G is placed, but here what we see, a new nucleotide is placed. So during this part, as a result of this addition of new nucleotide adenine here, it disrupts this open reading frame. How? Let's look at here. So previously there are 5 stretch of histidine that should be produced, but now this will stand for histidine. Again next 3 stand for histidine. Next 3, instead of a CAT it becomes ACA. CAT instead of ACA. So reading frame kind of change. Instead of CAT it becomes TCA, again completely different. Instead of CAT it is TCA again, right? And then it will go on like that in the future. Okay, so that thing happens. So from this level, you know, from the first 2 things, histidine and histidine are placed. But after that, ACA stands for adding threonine, so they will add threonine. TCA stands for serine, they will add serine, and again another serine. So you know, previously they should produce 5 histidines and they will produce proteins with this 5 histidine chain. But now they produce 2 histidine, threonine, serine, serine. So there is a remarkable different structure of the protein will be observed due to this Frameshift mutation. So it's not one particular change. Now I should compare this with silent mutation. You can see in silent or in missense or nonsense, whatever it is, there is a change in a particular nucleotide. It will change that one only, but rest of the nucleotide will remain same. But in case of silence it never does so, that's why it remains the same. But in case of missense or nonsense, there will be the change in the nucleotide, and what it does, it will change that nucleotide only, that amino acid only, and rest of the amino acid frame remains the same. But in this case, by adding one nucleotide extra, it will disrupt the whole open reading frame and it will produce a completely different type of proteins. So that's why Frameshift mutation is much more dangerous. It can produce much more variety of proteins inside the cell, right? This thing may happen also if we delete a nucleotide from that position. If we delete something from that position, it can also lead to the disruption of the reading frame. Until that deletion, everything will be the same. But whenever it is addition or deletion from the nucleotide, rest of the downstream nucleotide sequence will be changed, right? So that is the Frameshift mutation. So I hope you like this. If you enjoy this, please subscribe and watch future videos and all the videos regarding mutation. There is the link for the playlist, and other links will be given here up and down in different formats of annotations. So click to them, watch them, and if you want to learn more about these things, click and go to my website. They will find many things. Thank you.